Our last topic in this chapter is talking about how to collect the DNA evidence. So now that we've talked about some of the issues and what we're going to do with this DNA evidence and how I can use it, we can talk about, all right, so how does it affect how we collect it? One of the, we're going to have a variety of different sources of DNA. A lot of body fluids and body pieces from body can be used. So blood, so the fluids are particularly useful. So blood, semen, saliva are very good sources of DNA. Skin cells, we can get some from epithelial cells. We call this touch DNA because as you touch stuff, you might be a little bit, this sometimes cannot yield nuclear DNA, only mitochondrial DNA. Hair, unless you have the whole follicle and with the root to it, you probably can't get nuclear DNA, but you often can get mitochondrial DNA. And sometimes bone samples can be used. So there's a variety of different evidence that might be able to yield DNA evidence. So it is, we know, a very powerful creator of physical evidence. Now, especially that we have this PCR technique, so we can find DNA evidence on a lot of things and trace amounts. And now that we can amplify it and create more copies of it, this becomes pretty helpful. So bottles, glasses, things that you would have touched with your mouth, so bottles, cans, glasses, cigarettes, bite marks, envelopes are all great examples of having saliva-based evidence that could be used. Clothing, especially clothing that has blood evidence, should be collected. Dried blood can be used. However, with dried blood, the best way to collect it is to sort of take a wet cotton swab, sterile, of course, we'll talk about with gloves and all that, and then to sort of dampen it up and it sort of dissolves it and then onto the cotton swab. You must let the cotton swab dry for 5 to 10 minutes, and then you can put it into an envelope, and that's the best way to collect dried blood stains. Also, don't forget, we need a lot of standard and reference DNAs, right? So the easiest way to do DNA typing is to match two samples and say, yeah, these are identical. But that means that we need to have two samples. So we need to have a reference sample from, if it's a missing person, maybe from another family member. We need to have as suspects, we want their DNA and the DNA found at the crime scene. So it's important to have multiple samples if we're trying to do matching work, comparison work. When we package biological evidence, we've already talked about this. We're really concerned about making sure it's dry, but it's also important to really worry about contamination. This is because such trace amounts of DNA can be amplified. We have to be very, very careful about contamination. So again, we would first make sure that everything we had was photographed and recorded on sketches. And then we would begin with wearing just 2 layers of powder-free latex gloves. Powder-free means that we're not going to contaminate it. So the outer gloves are frequently changed in between samples. Also, you want to make sure you've got good protection, so hair nets, clothing, outside clothing on, so, we can have other things being contaminating. So it's really important when collecting DNA evidence that the collector is very clean. We would collect the entire clothing from the victim and the suspect that might have DNA on it. And of course, we're not going to put it into anything airtight, right? We need to make sure that we don't want to have mold growth or bacterial growth. So we need to make sure it's in something breathable, typically paper, right? So we've already talked about that, just repeating that information. Again, we're really concerned about this contamination because of this PCR amplifying. If the collector has any DNA added to the sample, that's going to get amplified and picked up as a contaminant. So double layered, powder-free gloves, face mask, lab coat, eye protection, shoe covers, hair covers, all of that. Every time you go to collect a new sample, changing the outer gloves to make sure there's no contamination. Collecting a substrate control, so that would mean like that your own DNA is on file so that they can at least eliminate that. If they do detect it, say, okay, but that belongs to the collector. Also, anything that's picking it up with disposable forceps. Typically, we would use plastic. We might be able to use forceps that could be re-cleaned. We talked about that in pate evidence. It's a little brittle, and so the little disposable ones aren't often strong enough. But in DNA, everyone gets a new pair of forceps, and they have to get thrown away. And then each item always is in its own evidence collection package, as usual.